Every single day, more than 2 billion people do something that feels completely ordinary. They take last night's leftovers, place them in the microwave or on the stovetop, heat them until they seem thoroughly warm, and eat them without a second thought. The assumption underneath this habit is simple and feels reasonable. If food is heated thoroughly enough, it must be safe to eat. That assumption is costing lives. Here's a number worth remembering. 1 in 6. That is the proportion of all cancer cases worldwide that researchers now associate with dietary factors, including the specific way that food is stored, reheated, and consumed. According to data published in CA, a leading cancer journal for clinicians, poor dietary habits rank as the second leading modifiable risk factor for cancer worldwide, sitting just behind tobacco use. And yet, almost no one is talking about what happens to certain foods at the molecular level when they are reheated for a second time. The chemical reactions that take place during reheating are completely silent. You cannot see them. You cannot smell them. You receive no warning that anything unusual is happening, but they are occurring inside your food and then inside your body every time certain foods go through the reheating process. What follows is a careful, evidence-based examination of 4 foods that you are almost certainly reheating. Foods that are probably sitting in your refrigerator right now. Foods that undergo documented, scientifically verified chemical transformations when subjected to a second round of heat. Transformations that carry measurable cancer risk. Stay with this through to the 4th food, because the 4th is one that almost every health-conscious person believes is completely safe to reheat. That belief is dangerously mistaken, and the science explaining why will change how you think about one of the most nutritionally celebrated foods in the modern diet. Before examining the specific foods, it is essential to understand what is actually happening chemically when food gets reheated. This is not optional background information. It is the key that makes everything else comprehensible. This is not about a minor inconvenience. This is not about an upset stomach or temporary food poisoning that resolves in 24 hours. What we are discussing is the slow, cumulative buildup of carcinogenic compounds inside your body across months and years of repeated exposure, the kind of silent, invisible damage that often goes completely unnoticed until it has progressed to a point that is extremely difficult to reverse. Cancer rarely develops overnight. It is a gradual process, building through accumulated biological events over time. The building blocks of that process, free radicals, advanced glycation end products, nitrosamines, and lipid peroxidation byproducts, often enter the body not through cigarette smoke or industrial pollution, but through an ordinary kitchen, through the reheating habits of people who believe they are eating carefully. The World Health Organization has classified several compounds formed during the reheating of certain foods as Group 1 or Group 2A carcinogens. Group 1 means there is conclusive evidence that the substance causes cancer in humans. Group 2A means it is probably carcinogenic to humans. These are not theoretical classifications based on preliminary observations. They reflect decades of scientific research and rigorous evidence review. And these compounds are being generated in ordinary kitchens, on ordinary stovetops, and inside ordinary microwave ovens every single day. The people at greatest risk are not those with unusual eating habits. They are ordinary people who believe they are making healthy food choices. Lean proteins, cooked vegetables, rice, eggs. These are the 4 food categories under examination today. To understand why reheating creates such different conditions from initial cooking, 3 fundamental biochemical mechanisms need to be understood. These 3 mechanisms appear repeatedly across all 4 foods, in different combinations and through different pathways. The first mechanism is oxidation. Fats and proteins that have been exposed to oxygen during storage in the refrigerator become chemically unstable. When heat is applied again to these already altered molecules, they break down further into compounds known as lipid peroxides and malondialdehyde. Both are well-documented agents capable of damaging DNA. The second mechanism is the Maillard reaction pushed beyond its safe boundaries. In normal first cooking, the Maillard reaction produces the appealing brown color and rich flavor of many cooked foods, a desirable outcome. However, when proteins and sugars are repeatedly exposed to high temperatures, they can produce compounds known as advanced glycation end products, abbreviated as AGEs. These compounds actively promote chronic inflammation, which is one of the primary biological drivers of cancer development at the cellular level. The third mechanism is nitrite conversion. Certain foods, particularly those naturally high in nitrates, undergo bacterial conversion while sitting in the refrigerator between the first cooking and the reheating. When these foods are subsequently reheated, the nitrates that bacterial activity has already converted into nitrites can be further transformed by heat into nitrosamines. The World Health Organization classifies nitrosamines as probable or confirmed carcinogens. Three mechanisms: oxidation, advanced glycation, and nitrosamine formation. These are the recurring themes across every food examined here. Understanding them transforms what follows from a list of warnings into a comprehensible map of specific biological risks. The first food is cooked rice. Rice is the most widely consumed staple food on Earth. More than 3 billion people rely on rice as their primary caloric source. Billions of those people reheat leftover rice every single day without giving it any particular thought. It is deeply ingrained as a habit across nearly every culture worldwide. But there is a serious and poorly understood problem with this practice, one that has less to do with the reheating itself and more to do with what happens during the time the rice spends in storage between the first cooking and the second. As cooked rice cools, it becomes an almost ideally hospitable environment for the growth of a bacterium called Bacillus cereus. This is not a rare or unusual contaminant. It is naturally present in raw rice, and it survives the initial cooking process. Once the rice cools, Bacillus cereus can multiply rapidly, whether the rice is left at room temperature or placed in the refrigerator. The critical point is this: when the rice is reheated, the heat may kill the bacteria themselves, but it does not destroy the heat-resistant toxins those bacteria have already produced and deposited in the food. Those toxins survive the microwave. They survive the stovetop. They enter the digestive system intact and active. The bacterial contamination risk is significant and real on its own, but the cancer concern goes deeper than food poisoning. Bacillus cereus produces a toxin called cerulide. Laboratory studies have demonstrated that cerulide can damage the mitochondria inside human cells. Mitochondrial dysfunction is recognized as one of the early biological events that can contribute to malignant cellular transformation, the process by which a healthy cell gradually becomes cancerous over time. There is a second, less frequently discussed but equally important concern associated with reheated rice. Repeated heating of starch-rich foods increases the formation of acrylamide, a chemical compound produced when natural sugars react with the amino acid asparagine under conditions of high heat. The International Agency for Research on Cancer classifies acrylamide as a group 2A carcinogen, probably carcinogenic to humans. This classification is based on a substantial body of animal and human research. What should you do instead? Cook rice fresh whenever possible. When storage is necessary, spread the cooked rice out on a shallow tray immediately after cooking so it cools rapidly rather than slowly. Refrigerate it within 1 hour of cooking. Consume refrigerated rice within 24 hours, and never reheat rice more than once. A single reheating, consuming it promptly, and then discarding any remaining portions is the maximum safe handling for stored cooked rice. The second food is cooked chicken. This one may be more surprising because chicken occupies a uniquely trusted position in modern health-conscious eating. Chicken is the world's most popular lean protein. It is recommended by doctors, dietitians, fitness professionals, and nutritional guidelines across essentially every culture and food tradition. When leftover chicken ends up in the refrigerator, which it does in tens of millions of households every day, almost everyone reheats it. The idea that this common, widely celebrated practice carries documented cancer risk is not something that most people have ever encountered. Here's the biological mechanism. When proteins in animal muscle tissue are exposed to high temperatures for a second time, they undergo a process called protein oxidation. One significant consequence of this process is the formation of compounds called heterocyclic amines, abbreviated as HCAs. Heterocyclic amines are not a controversial or contested topic in the scientific literature. They are among the most extensively studied dietary carcinogens in modern nutrition science. Once HCAs enter the body, liver enzymes metabolize them into highly reactive compounds that can bind directly to DNA. That direct interaction between these compounds and DNA can produce genetic mutations. When those mutations affect critical genes, particularly tumor suppressor genes, which normally function to prevent uncontrolled cellular growth, they represent one of the primary molecular mechanisms through which cancer begins at the cellular level. A landmark study published in the journal Carcinogenesis found that individuals who frequently consumed well-done or reheated poultry had a statistically significant increased risk of colorectal cancer. A separate analysis conducted by the Harvard T.H. Chan School of Public Health found that frequent consumption of reheated processed chicken was associated with an increased risk of pancreatic cancer, one of the most deadly forms of cancer, with a 5-year survival rate of only approximately 11%. Lipid oxidation also occurs during the reheating of chicken. The fats present in chicken tissue, having already undergone initial oxidation during the first cooking and then additional oxidative changes during refrigerated storage, produce secondary oxidation byproducts, including malondialdehyde and 4-hydroxynonenal when subjected to high heat again. Both of these compounds have been documented in scientific literature as agents capable of damaging cellular DNA and promoting inflammation. The practical guidance here is straightforward. Whenever possible, eat leftover cooked chicken cold rather than reheated, incorporated into salads, wraps, or other dishes that do not require a second round of heat exposure. When reheating is genuinely necessary, use the lowest effective temperature and avoid overheating beyond what is required to reach a safe internal temperature of 165 degrees Fahrenheit. Never reheat chicken more than once, and avoid the common meal prep pattern of cooking large batches of chicken on a single day and then reheating portions repeatedly throughout the week, because each reheating cycle adds another layer of protein oxidation and HCA formation to what has already accumulated from previous heatings. The third food is one that will particularly concern people who take pride in eating a clean, nutrient-dense diet. It is spinach and other nitrate-rich leafy greens. Spinach is arguably the most celebrated vegetable in health-conscious eating. It is genuinely extraordinary from a nutritional standpoint, rich in iron, folate, magnesium, vitamin K, and a variety of powerful antioxidants. Nutritionists, cardiologists, and oncologists consistently recommend eating more spinach, and they are absolutely right to do so. When spinach is eaten fresh or cooked once and consumed immediately, it delivers exceptional nutritional value with negligible risk. The problem arises specifically when cooked spinach is refrigerated and then reheated. Spinach, along with celery, beets, lettuce, and Swiss chard, is among the richest dietary sources of naturally occurring nitrates. In their original form, these nitrates are not only harmless, but actively beneficial, supporting cardiovascular function and contributing to healthy blood pressure regulation. The nitrates themselves are not the problem. The problem is what happens to those nitrates during refrigerated storage. Once cooked spinach is placed in the refrigerator, naturally occurring bacteria present on the surface of the leaves begin converting the nitrates into nitrites. This is the same chemical conversion that occurs in processed and cured meats. It is one of the primary reasons the World Health Organization classified processed meat as a Group 1 carcinogen in 2015, meaning there is conclusive evidence that it causes cancer in humans. When spinach that has been refrigerated for 24 hours or longer is subsequently reheated, you are applying heat to a vegetable that has already undergone substantial nitrate-to-nitrite conversion during storage. Under the conditions created by reheating, those nitrites can react with amino acids present both in the spinach itself and later in the stomach to produce nitrosamines. Nitrosamines are among the most potent dietary carcinogens that modern science has identified. Animal studies have demonstrated that they can induce cancer at remarkably low doses. Human epidemiological research consistently associates high dietary nitrosamine exposure with increased risk of gastric cancer, esophageal cancer, and colorectal cancer. This is not a hypothetical concern or a theoretical extrapolation from animal data. The European Food Safety Authority has issued formal official guidance specifically addressing dietary exposure to nitrates and nitrites from vegetables, with explicit warnings about the potential risks associated with reheating nitrate-rich vegetables after they have been stored following cooking. The irony embedded in this finding is striking and worth sitting with. One of the genuinely healthiest foods available, a vegetable that nutritional experts enthusiastically encourage people to eat in greater quantities, can become a source of potent carcinogenic compounds through a storage and reheating habit that millions of health-conscious people practice every week, without any awareness that they are doing something that undermines the very health goals they are pursuing. The practical guidance for spinach and other leafy greens is clear. Cook them fresh and eat them immediately whenever this is possible. If you have prepared cooked spinach and cannot eat it immediately, eat it cold within a few hours rather than refrigerating it with the intention of reheating it later. If storage is unavoidable, keep it in an airtight container, consume it within 6 hours, and eat it cold rather than reheating it. Never refrigerate cooked spinach overnight with the plan of reheating it the following day. The combination of extended refrigerated storage and subsequent reheating is precisely the sequence that creates the most significant nitrosamine exposure risk. Now, the fourth food, the one that will genuinely surprise the people who have been most careful and most attentive about their nutritional choices: eggs. Eggs are among the most nutritionally complete foods in the human diet. They contain all 9 essential amino acids in proportions that closely match human nutritional requirements. They provide choline, an essential nutrient that supports brain function and cellular membrane integrity. They supply significant amounts of selenium, a mineral with important antioxidant roles in cellular defense. They contain lutein and zeaxanthin, 2 antioxidant compounds that protect both eye health and cellular health more broadly. By essentially every standard measure of nutritional science, eggs are one of the most beneficial foods a person can eat regularly, which is precisely why the reheating risk associated with eggs is both so important and so rarely discussed. People who eat eggs regularly for their excellent nutritional profile deserve to know what reheating does to those nutritional qualities, and what new compounds it creates in the process. Here is the mechanism. Eggs contain approximately 13% fat, with most of that fat concentrated in the yolk. When an egg is cooked for the first time, the fats in the yolk undergo an initial stage of oxidation, a normal, expected, and generally manageable process that occurs in essentially all fat-containing foods during cooking. The problem begins when a cooked egg is refrigerated and then reheated at high temperature. The fats that have already undergone initial oxidation during the first cooking and that have continued oxidizing during refrigerated storage as they are exposed to oxygen undergo secondary lipid oxidation at a much faster rate when heat is applied again. The byproducts produced during this secondary oxidation process, particularly malondialdehyde and oxysterols, are now well documented in the scientific literature as compounds of significant concern. Oxysterols deserve specific attention because they represent one of the more recent and more alarming findings in food chemistry research. Oxysterols are oxidized forms of cholesterol that develop specifically when foods containing cholesterol are repeatedly subjected to heat exposure. A comprehensive review published in the journal Food and Chemical Toxicology concluded that oxysterols possess pro-atherogenic properties, meaning they promote arterial disease, pro-inflammatory properties, and, most critically, cytotoxic and genotoxic effects on human cells. The word genotoxic is worth pausing on. Genotoxic means toxic to the genetic material inside cells, to DNA itself. By definition, genotoxic compounds are classified as potential carcinogens because they can produce DNA strand breaks and genetic mutations that initiate the molecular chain of events leading to cancer. When a compound is described as genotoxic in peer-reviewed scientific literature, The connection to cancer risk is not speculative. It is the expected biological consequence of that compound's mode of action. There is an additional concern associated with reheated eggs beyond the oxysterol problem. When cooked egg proteins are exposed to the high and often uneven heat produced by microwave reheating in particular, they undergo additional protein denaturation. These proteins form abnormal crosslinks that make them more difficult for the digestive system to break down effectively. More significantly, the sulfur-containing amino acids naturally present in eggs, cysteine and methionine, can degrade during repeated heating, and the degradation products formed from these amino acids may contribute to oxidative stress within the lining of the digestive tract. Persistent oxidative stress affecting the gastrointestinal mucosa, the cellular lining of the intestinal tract, is not a minor or incidental concern. It is considered one of the primary proposed mechanisms involved in the development of colorectal cancer, which is the third most common cancer worldwide and responsible for nearly 1 million deaths every year. Repeated reheating of eggs through the combined mechanisms of oxysterol formation and oxidative stress to the intestinal lining creates a specific and underappreciated pathway of dietary carcinogen exposure that accumulates silently with each reheating event. The practical guidance for eggs follows logically from the mechanism. Eggs are best prepared fresh and eaten immediately after cooking. If you prepare hard-boiled eggs in advance, store them with their shells intact. The shell provides a degree of protection against oxidative changes during storage, and eat them cold rather than reheating them. Avoid reheating scrambled eggs or omelets in the microwave, where uneven heating creates localized hotspots that accelerate the oxidation reactions producing oxysterols. If warming an egg-based dish is genuinely necessary, use gentle indirect heat at low temperatures rather than direct microwave or high stovetop heat. Avoid heating egg dishes to temperatures exceeding 165 degrees Fahrenheit. When all four foods are considered together, the pattern that emerges is important in what it reveals about both the scope of the problem and the simplicity of the solutions. Rice, with its bacterial toxin reactivation and acrylamide formation, represents a risk that billions of people create for themselves daily through a habit so ingrained that it is never questioned. Chicken, with its HCA formation through protein oxidation and secondary lipid oxidation byproducts, represents a risk that is especially perverse given chicken's position as the most widely recommended lean protein in health-conscious eating. The very food that fitness professionals and nutritional guidance celebrate becomes, through a second round of high heat, a source of compounds with documented direct DNA interactions. Spinach and other leafy greens, with their nitrate to nitrite to nitrosamine conversion pathway, represent a risk that is particularly cruel in its irony. The people most committed to healthy eating, who have specifically incorporated more vegetables into their diet in response to nutritional guidance, are the people most likely to be preparing cooked leafy greens ahead of time and reheating them throughout the week as part of a planned approach to healthy eating. Eggs, with their oxysterol formation and contribution to intestinal oxidative stress, represent perhaps the most overlooked of the four risks, partly because eggs' exceptional nutritional profile creates a default assumption that they can do no wrong, and partly because the specific mechanism, cholesterol oxidation through repeated heat exposure, is not part of the public discourse around egg safety in the way that bacterial contamination concerns are. The thread connecting all four is the central insight of this entire examination. The first cooking changes food in ways that make a second round of heat more chemically consequential than the first. Molecules that have already undergone oxidative changes become more reactive when heat is applied again. Nitrates that bacterial activity has already converted to nitrites become nitrosamines when reheated. Proteins that have already unfolded and cross-linked form more abnormal structures when denatured again. The second heating is not a repetition of the first. It is a chemically distinct event with chemically distinct consequences. This insight has immediate practical implications that require no special equipment, no expensive supplements, and no drastic lifestyle changes. The solutions are genuinely simple once the problem is clearly understood. Cook rice fresh whenever possible. When storage is necessary, cool it rapidly, refrigerate it within 1 hour, consume it within 24 hours, and reheat it only once. After a single reheating, discard any remaining portions. Eat leftover chicken cold whenever possible. When reheating is necessary, use the lowest effective temperature and never reheat chicken more than once. Avoid the common meal prep pattern of repeated reheating throughout the week. Do not reheat cooked spinach, Swiss chard, beet greens, celery, or other nitrate-rich leafy greens. Eat them immediately after cooking, or eat them cold within a few hours of cooking. 6 hours is the outer limit for cold consumption of cooked leafy greens. After that, discard them rather than reheating them. Cook eggs fresh and eat them immediately. If preparing hard-boiled eggs in advance, store them unpeeled and eat them cold. Avoid microwaving scrambled eggs or any egg-based dishes. Use gentle, low-temperature heating if warming an egg dish is unavoidable. Apply the universal principle across all foods: one cooking, one reheating, maximum. Beyond that single cycle, the cumulative chemical changes that have occurred in the food make additional reheating inadvisable from a cancer risk perspective, regardless of the specific food involved. Invest in a food thermometer. Gentle reheating that brings food to a safe temperature without exceeding it, staying below 165 degrees Fahrenheit rather than heating food aggressively beyond that threshold, can substantially reduce the rate of many of the oxidation reactions discussed above. Use a low-temperature oven instead of a microwave whenever reheating protein-rich foods. Microwave ovens create uneven, localized hotspots within food that can reach temperatures substantially higher than the average temperature reading, accelerating oxidation reactions in specific areas of the food, even when the overall temperature seems controlled. Plan meal portions carefully to minimize leftover production in the first place. This is the single most effective behavioral adjustment available. If fewer leftovers are produced, fewer reheating decisions need to be made. The frustration underneath all of this information is real and worth acknowledging. None of the research that underlies these findings is hidden or obscure. It has been published in peer-reviewed scientific journals. It appears in formal guidance from the European Food Safety Authority, the World Health Organization, and numerous public health agencies around the world. It has been available in the scientific literature for years, and yet it almost never reaches the dinner table in a form that allows ordinary people to act on it. There are children eating reheated rice, spinach, and eggs in their daily school lunches with no awareness that the reheating habit is creating compounds that would not have been present in those foods when they were freshly prepared. There are older adults, particularly those living alone for whom batch cooking and reheating is a practical necessity, who are reheating leftover chicken in the microwave every evening without any information that would allow them to make different choices. There are fitness-conscious people eating reheated high-protein meal prep containers every day, convinced that they are maximizing their nutritional investment, unaware that the chemical composition of their carefully selected foods has shifted significantly from what it was when originally prepared. None of these people are being careless or uninformed through any fault of their own. They have simply never been given this specific information in a form that was clear, practical, and accessible. The gap between what nutritional biochemistry has documented and what ordinary people know about their own food handling is one of the most consequential information gaps in preventive medicine. The purpose of bringing this information forward is not to create anxiety about eating or to suggest that occasional reheating of these foods will produce immediate or certain harm. The relationship between dietary carcinogen exposure and cancer development is one of cumulative risk over time, not of a single meal determining a health outcome. The concern is with repeated habitual exposure across years, the gradual accumulation of carcinogenic compound exposure that comes from reheating these specific foods as a default daily practice without awareness of the chemical consequences. A single reheated plate of chicken or spinach does not cause cancer. A decade of reheating these foods multiple times each week, combined with all the other sources of oxidative stress and carcinogenic exposure that modern life involves, contributes meaningfully to cumulative risk in ways that the science is clear about, even if public awareness has not caught up with it. Making simple, informed changes to daily food handling habits, cooking fresh more often, eating leftovers cold rather than reheated when possible, following the one reheat maximum rule consistently, and being particularly careful with the 4 specific foods identified here, costs nothing and requires no medical intervention. It simply requires knowing what the science has established and applying that knowledge to decisions that are already being made every day anyway. The decision about how to handle leftovers happens multiple times each day for most people. Making that decision with accurate information about what reheating does to specific foods at the molecular level is simply preventive medicine in its most practical and accessible form. Everything presented here reflects findings from peer-reviewed scientific literature. Food science is an active field, and understanding continues to develop as new research accumulates. The core findings regarding nitrosamine formation in reheated leafy greens, HCA formation in reheated proteins, oxystrol development in reheated egg yolks, and bacterial toxin activity in reheated rice are not preliminary or contested findings. They are well-established results from multiple independent lines of research that have been replicated across different laboratory and epidemiological methodologies. This information belongs at the dinner table. It belongs in the conversation that happens before someone puts last night's chicken and spinach back in the microwave. It belongs with every person who has ever assumed that thorough heating is the only requirement for leftover food safety. Now you have it. What you do with it is a simple matter of applying knowledge you did not have before to decisions you were already making daily. The research is real, the mechanisms are documented, and the practical changes required to substantially reduce this specific category of dietary cancer risk are among the most straightforward and accessible in all of preventive nutrition. One cooking, one reheating at most, fresh whenever possible, cold when reheating is not an option. That is the evidence-based framework, and it is available to anyone willing to apply it.